Hello, everybody, and welcome to our, I think it's our 10th meeting. And we're going to have Viviane Quirke, the well-known historian of pharmacy from book Oxford Books University. And we're going to be talking to us about the ICI and the molecularization of science, how basically modern pharmaceuticals started in Cheshire back in the 1950s. So over to you, Viviane.
Okay, thank you. Thank you very much, Peter, for the invitation. It's lovely to be back and see some familiar names. So I'm going to talk to you about two of ICI's many research programs, one on cardiovascular diseases, which led to the first clinically and commercially useful, successful beta blockers, and the other one, the cancer research program, which produced tamoxifen, which later became known as one of the first selective estrogen receptor modulators. And this is partly my choice due to the context we're living through at the moment, the context of the COVID pandemic, of course. We, the public, we've become used to seeing certain images on our screens and in newspapers, images like the one on the left, which represents the virus COVID-19. And what's often been talked about and highlighted is the spike protein, which vaccines are trying to block, but and drugs as well, and which are being changed in various mutations of the virus, thereby escaping both vaccines and potentially drugs as well. And the way we've tried to combat the virus is not only through vaccines, which are represented on the right with a syringe pointing precisely at the spike protein on the surface of the virus, but also various drugs, amongst them anti-inflammatories such as dexamethasone, hydroxychloroquine's been mentioned too, which is related to an anti-malarial drug, azithromycin, common used antibiotic, of course, antivirals like remdesivir, and monoclonal antibodies. And I found the image of monoclonal antibody action against COVID most illustrative of the type of activity targeting the spike protein on the surface of the cell. And the interaction between that protein and the host cell is shown on the left-hand side, and that's what the antibodies are trying to block and prevent, thereby limiting the severity of the disease. So how did that sort of understanding of disease developed? It has a history, and that's the sort of thing I've been interested in. The history is being referred to as a process of molecularization, and historians of medicine have argued that through that process and the biomedical approach, which has made drugs the mainstay of medical practice today, drugs that embody that sort of action against disease, have deprived people of some of the benefits of holistic medicine, which I represent on the right, with the doctor sitting by the patient's bedside and considering not just his illness, but his whole illness located in a particular organ, but his whole body, and even his lifestyle, his life setting. So what I want to do in my talk is highlight the part played by ICIs, drugs, more particularly the beta blockers and tamoxifen. And in doing that, I want to talk about the process of molecularization and how it came about historically. So the complex relationship that produced it, the complex relationship between drugs, their action in the body, theories of drug action, how they get played out in R&D, and the feedback loops that this represents. It's a much more complex process than a simple linear process, as the word molecularization might lead to suggest. It's a process of feedback loops, as represented in this image on the right-hand side, which I found on the internet. It's out of a Nature Reviews drug discovery article. It shows the starting point, which is for drug development, which is a hypothesis, design, then of a molecule, linked to chemical intuition, various approaches which are used today, like combinatorial approaches, molecular modeling, de novo methods. In the period that I'll be talking about, these hadn't come to fruition yet. But nevertheless, the model, which is represented here, very much fits both the history of the beta blockers and the tamoxifen, and I'll hope to convince you of that, because before the drugs can be put into the body, they have to be tested, and then it doesn't end there. Once they are in the body, they feed back information, which can lead to changes, new hypotheses, and changes in the types of molecules that are designed to fit these observations in the clinic. So the model on the right is what might be understood to be in place today, but it wasn't always like that. It has evolved and changed over time, and I'll be talking a bit about how that happened. So the plan for my talk is I'll talk about first the cardiovascular disease program and how it led to propranolol, or Inderal as it was marketed. Then I'll talk briefly about the cancer research program, which led to tamoxifen, although it was intended initially as a contraceptive pill. So it was the product of a double program to look for a contraceptive pill and work on breast cancer at the same time. And this will lead me to some concluding remarks about the relationship between theory and practice in pharmaceutical innovation and the role of ICI, which now we would know as Zeneca, as in AstraZeneca, then and now. Of course, it won't have escaped you that AstraZeneca produced one of the vaccines that are the workhorses of the vaccination programs around the world today, in collaboration, of course, with the Jenner Institute at Oxford University. First of all, a few words about ICI's pharmaceutical division. It was the outgrowth of the medicinal chemistry section of the dyestuffs division, which was situated at Blakely, outside of Manchester. At the end of the Second World War, it's split off into its own division, the pharmaceutical division, but it was still, the research was still being done at Blakely. And in many ways, the pharmaceutical division was still under the auspices, still came under the auspices of the dyestuffs division. Changes started to take place in the growing independence of the pharmaceutical division and the R&D that was being carried out within it in 1948, with the creation of a chemotherapeutic research committee, which set off the research program for the pharmaceutical division. And by 1953, this committee was making plans for a center for speculative research, which would become Alderley Park. And Alderley Park, south of Manchester in Cheshire, was opened in 1957. That's where much of the research I'll be talking about actually took place. Now, a few words about what was meant by speculative research at the time. It was a term used which I found in the archives. It was meant, by that was meant research based on scientific hypotheses, whichever hypotheses were available at the time, with structure-activity relationships helping to connect these scientific hypotheses to the synthesis of molecules of hopefully future drugs. However, it was not without its skeptics. There were some people who thought that, especially when it became very quantitative, it was quite an esoteric route down which to go compared to more traditional synthetic organic chemistry. But I suppose that's the way history of R&D takes place, with skeptics, debates, controversies, and these being gradually eroded over time, just as combinatorial chemistry, I believe, has its skeptics today. From the beginning of the Chemotherapeutic Research Committee and later on Alderley Park, hypertension, rather than cardiovascular diseases as a whole, was identified as a potential target. And it became a team project in 1954. This organization in team projects was very important for the future of the pharmaceutical division. And it was quite novel at a time. It was something I think that some American companies who did that did R&D had. Well, ICI was one of the first companies to adopt this sort of team project approach, certainly in the pharmaceutical field in Britain. So hypertension became a team project for the new pharmacology section that was created in '54 under Alfred Spinks, who later on became very well known. He rose through the ranks of the company, and he wrote the Spinks report for Margaret Thatcher on biotechnology. In 1955, there was an addition to the pharmacology section with a project on atherosclerosis, which led to clofibrate under the leadership of Jeff Thorpe. And the work of that section produced by 1958 a ganglion blocker for the treatment of hypertension called pempidine. But there were problems with these ganglion blockers. They often led to some unpleasant side effects, so they were hard to use in the clinic. Amongst the side effects were postural hypertension and constipation. Therefore, by '58, the company was thinking about changing its direction in relation to hypertension and thinking more broadly about cardiovascular diseases as a whole. And that's when James Black arrived at just the right time. He was the right person in the right place at the right time, both for ICI and for the history of drug development. James Black was interested in looking for a new type of drug to treat angina pectoris. And his approach was based on a theory, as it was at the time, a cutting edge theory, which was Raymond Alquist's dual alpha and beta receptor theory. A bit more about this theory. You've probably come across the theory mostly in connection with Paul Ehrlich. Ehrlich has been written about expertly by Anthony Travis, who's shown that his ideas about drug action were very much closely connected to his experience with dyestuffs. It was also connected to his experience with antitoxins, in particular the diphtheria antitoxin. And he was looking for a way of selectively targeting pathogens and thinking about the magic bullet, if you like, as a mirror image of the body's immune response. He postulated that immune responses were due to molecules extending from cells, so a chemical structure which he referred to as side chain, containing receptors. He was one of the first to use the term receptors, although the idea of a side chain had been developed almost at the same time by the British physiologist John Newport Langley, who believed that these receptive substances, he didn't use the term receptor, mediated the action of a drug in a cell. The research that followed Langley and Paul Ehrlich's seminal work is too complex, and I won't go into detail here, but I will just point out that there was a greater belief in the whole receptor concept after A.J. Clark's, another British physiologist, actually was Scottish, I believe, in 1933, when he published his mode of action of drugs on cells. He explained that the pharmacological action produced by drugs was directly proportional to the number of receptors occupied. And there followed a number of other work which eventually led to Raymond Alquist's dual receptor theory, which would be very important for the development of ICI's beta blockers. In 1948, Raymond Alquist, who was an American pharmacologist, explained that in the heart, excitatory responses to compounds like isoprenaline, an analog of adrenaline, such as the increase in rate and force of contraction, corresponded to the beta blockers and not the alpha receptors as in other tissues. That helped to explain the fact that the same compounds could have different effects, different types of effects on different types of tissues. And it was this idea that James Black came across in Drill's Pharmacology in 1954. And it gave him the idea that it should be possible to treat angina pectoris by blocking the beta receptors to the heart using compounds like isoprenaline, instead of trying to increase the supply of oxygen to the heart, as was traditionally done, but with some unpleasant and unsatisfactory side effects. So by 58, having developed his ideas and wanting to do more research on it, he turned to ICI for a research grant. He knew of somebody who was a sales rep for ICI, and he talked to him about his ideas, and the sales rep said, why don't you turn to ICI? And Black impressed ICI so much that he was offered a position at Alderley Park, which had just opened. And Black, having heard of this sort of fairy tale place, as he described it, that Alderley Park was, as a research lab in Britain at that time, he promptly accepted. And he was very much left free at Alderley Park to do the research he wanted, even though it was quite an unusual research project. It hadn't really been done that before. ICI very much left him alone, partly because I think at that time the pharmaceutical section was still fairly marginal and below the radar of the bulk of ICI, and helped by a group of ICI chemists, but also technicians. The first true beta blocker was synthesized by ICI in 1960. It was ICI 38,174, which would become known as prenetholol or aldolin. And Stevenson, who was the chemist who helped Black, it's useful to mention, used a chemical structure activity approach to improve on compounds that already existed and were known to have the sort of desirable action that, that Black was looking for. Now, once prenetholol had been launched, of course, it had been patented. The patents needed to be protected, and more research had to be done around prenetholol to try and protect the product against potential competitors. Pharmaceutical companies were always watching what each other did. And so any developments like this were closely followed by others. In the process of doing this research, which was much less interesting to Black, but still, you know, he worked in a pharmaceutical company. He had to do what the company expected of him. He started to think about Alquist's dual receptor theory. And he started thinking that maybe it was more complex than that. There were some receptor subdivisions, not just alpha and beta or beta, but beta 1, beta 2. And this suggested to him that it might be possible to develop compounds which had even more selective blocking activity on the beta blockers of the heart than prenetholol. He became fascinated with the idea, and he thought he'd like to do the same with gastric secretions for the treatment of peptic ulcers. So he asked ICI if he could work on this area, but ICI were still building on the research and development of prenetholol, and they discouraged him. Therefore, Black left. He actually left ICI for Smith, Klein and French to work on histamine antagonists, which would lead to the first H2 blockers in use in the world, really, as treatments for peptic ulcers. Meanwhile, amongst the numerous compounds that were synthesized as part of the patenting and sort of looking for further maybe successors to prenetholol, ICI 45520 had been synthesized, and it was selected amongst numerous analogues, as you can tell from the number 45,520, as a potential successor for prenetholol, which would be more potent, but also would have fewer side effects. By 1969, the Committee for the Safety of Medicines gave clearance for propranolol, now which was referred to as Inderal, an anagram of Alderlin, which was the name given to pronetholol after Alderley Park, and it was cleared specifically for hypertension. Now, this happened at the same time as the beta receptor protein was isolated from cell membranes. Of course, there were all kinds of technical developments that occurred to make this possible, which I can't go into detail, but it happened at just the right time, because the drugs would then become not just treatments for specific diseases, but research tools in research surrounding the receptors. And we'll see this story repeated with tamoxifen. And it kicked off what's been referred to as the age of the receptor. Following the beta blockers, Alquist dual receptor theory was applied in pulmonary medicine, obstetrics, endocrinology, gastroenterology, and psychiatry as well. And hence, following Black's research at ICI, one can really trace it to that moment. The 60s and 70s has become known as the age of the receptor. Now, if you don't, I mean, just to give you an idea of how the drugs themselves could become tools for research into the receptors, the ICI chemists, who were brilliant, as no doubt you will know, prepared a beta blocker with a fluorescent probe attached to it for use as a tool in the investigation of the beta receptor. So I've seen in the archives clear demonstration that there was this back and forth between the drugs as treatments and the drugs as research tools, the drugs as treatments in the clinic and back into the lab as research tools. The problem with Inderal, however, was that as it was being used in the clinic, it was found that although it relaxed the muscles of the heart, it had a constrictive effect on the lungs, and therefore it was counterindicated for asthma sufferers. The search for beta blockers that would be cardioselective, therefore, continued even as Inderal was being used in the clinic, but only for people who didn't suffer from asthma. In 1970, Practolol was developed for those asthma sufferers who still needed the benefits of a beta blocker. And that was approved by the Committee for the Safety of Medicines. However, it had deleterious effects in some people. It had some of these very rare side effects, which you'll think again, history seems to repeat itself. It had an effect which was just seen in the population in connection with Iraldin and nothing else. Rare, I think, oculomuco syndrome, which affected the eyes and could lead to blindness, and in some cases it could even lead to death. Therefore, by 75, it was Iraldin. Iraldin was removed. It was withdrawn from the market. A compensation scheme by ICI was set up to compensate those who'd suffered or the families of those who died as a result of taking Iraldin, and a successor was already waiting in the wings in the shape of atenolol or tenormin, which by the 1980s was so successful it made 500 million for the company. Now I move on to ICI's cancer research project, which coexisted with the cardiovascular project, and sometimes the chemists worked on both projects, some specialists in particular technologies would help with both projects. And so in the context of a company, more so perhaps than a university, the walls between projects and between disciplines are quite permeable. Nevertheless, the cancer research project evolved on its own, separately from the cardiovascular project. And apart from a few chemists, the biologists certainly worked separately on the cancer research project. They didn't work on the cardiovascular research project. And I've represented the evolution, which is quite complex, on this slide with some arrows. Now, just a few words. The cancer research project really starts in the late 1930s with the finding that triphenylethylene, which was an analog of stilbestrol, had estrogenic properties and a chemical structure that resembled estrogen. And that finding was made at a time when there was a growing understanding that certain cancers, in particular breast cancer, were hormone dependent, so they were connected to estrogen, of course, without any ideas of estrogen receptors having been developed at that time. The cancer research or interest in cancer wasn't really a project at that time. ICI's interest in cancer continued through the war with work on nitrogen mustard, which, as you know, was found to shrink tumors in an American lab. And because of Anglo-American collaboration, this research was picked up on this side of the Atlantic. After the war, cancer continued to be a subject of interest, and it became a proper project in '55 with under the label Cancer and Viruses. It was around the time that interferon was being discovered and thought about in terms of a treatment against cancer. But eventually the topic split off and contraception, cancer went off on its own, merging with a contraception project, whilst viruses were being studied on their own. And tamoxifen was the result of that cancer and contraception project once it had become the endocrinology and fertility project. So tamoxifen 46,474, ICI 46,474, was synthesized in 1962 as part of this contraception endocrinology project, and it was intended to be a contraceptive pill. It was synthesized by the female chemist, ICI chemist Dora Richardson. And what's often described in terms of her synthesis is not just how a feat the synthesis itself was, but she managed to identify in which isomer of tamoxifen the activity resided. And she did this by a process of fractional crystallization. So it was considered to be quite a feat at that time. Now, tamoxifen didn't come out of the blue. It was a derivative of triphenylethylene with groups and side chains to enhance its anti-estrogenic and pituitary inhibitory effect and prolong its duration of action, whilst not interfering with its fertility properties as it was intended for a contraceptive pill. But the leader of the project, Arthur Walpole, had long been interested, he was a biologist who'd long been interested in cancer as well as endocrinology, and therefore he insisted in cancer being included in the patent that was filed for tamoxifen in 1963. A few years later, in the wake of the isolation of the beta blocker protein, the estrogen receptor protein was also isolated by a group in America. Although this didn't have an immediate impact on ICI's research project. In 1969, the Committee for the Safety of Medicines approved Nolvadex in clinical trials, eventually both in contraception and in cancer, but already there was a conundrum. It seems that at high doses, tamoxifen stimulated rather than suppressing ovulation, and at low doses, it had anti-estrogenic effects, so it produced hot flushes, and it also appeared to shrink breast tumors. So how could that be? How could both be? That puzzle wasn't solved straight away. First of all, it had to be used in the clinic. Its application in the clinic was almost a sine qua non for research in the lab, as tissue samples from the clinic could be used for research in the lab. And this would lead to the next phase in the development, but we're not there yet. In 1970, the development department at ICI thought that, well, if it, as a fertility drug, if that's what it was going to turn out to be, and as a breast cancer treatment, it would make limited sales that would barely cover the R&D costs of ICI. They would represent 100,000. And Richardson, in her history of the program, said obviously the development department only considered treating dead people to have such low sales estimates. And the development department, because of these low sales estimates, put a lot of pressure on the company to close the development program, and it was nearly stopped. But Walpole, the leader of the project, who'd been with the company since 1938, threatened to resign. So the company reversed its trajectory, and the program was approved, and the rest is history. Walpole promptly set out to devise an estrogen receptor protein binding assay method in order to start making use of these tissues that could be obtained from the clinic. And a simpler method was developed in collaboration with Craig Jordan, who was then on leave from Leeds at the Worcester Foundation. This work led to tamoxifen's mode of action being elucidated, or at least partly elucidated, when it showed that tamoxifen had a high affinity to the estrogen receptor, but low efficacy, so it was a partial agonist. And this made it a very useful tool for research as well as treatment, research into hormone-dependent tumors, and for predicting the response of breast tumors to hormone therapy. In the process of doing this research, the Committee for the Safety of Medicines approved its use in both anovulatory infertility and breast cancer in the UK. And by 1976, it was approved for breast cancer in the US. Now, here there's a quite stark contrast between the cardiovascular story and the tamoxifen story, the Inderal story and the tamoxifen story. ICI became highly dependent on an external lab. In fact, it very much outsourced its research from 1980 onwards, at least, to an outside lab. Craig Jordan's tamoxifen team at Leeds was funded by the Yorkshire Campaign Fund and an ICI Leeds Joint Research Scheme. Twice a year, Jordan's team went over to Alderley Park to present their findings, and he reported that tamoxifen could prevent rat mammary carcinoma, longer adjuvant, so on top of surgery, adjuvant tamoxifen therapy was better than shorter 1 to 2 years adjuvant tamoxifen therapy, and only patients with estrogen receptor positive tumors would benefit from tamoxifen therapy. By 1980, the sales had been bumped up to 30 million, so quite a stark contrast from the 100,000 predicted by the development team. In the same year, there was no longer any research program at ICI, however, so it's a sort of paradox, because Walpole had died suddenly in 1977, and the Cell Division and Growth Project had ended after its lead, Stephen Carter, retired. So the reliance on Craig's lab became even greater. He led a number of clinical trials where he, from different bases, now over in the US, where he continued to receive funding from ICI. One of the trials he led was the breast cancer prevention trial, which helped to establish tamoxifen as the first chemopreventive remedy for cancer. And later on in the 90s, he also led the STAR trial, which compared tamoxifen to second generation SERM selective estrogen receptor modulators, as tamoxifen was now understood to be. And this helped to establish the principle of SERMs more widely. But again, in a way that's almost a repetition of the cardiovascular story, AstraZeneca, by now, chose not to pursue SERMs, but aromatase inhibitors instead, and the company and Jordan parted ways. In fact, Jordan became a consultant no longer for AstraZeneca, but for Eli Lilly, which made a fortune from SERMs. It didn't stop, of course, ICI from making a fortune from spin-offs of tamoxifen, Casodex, I think Zoladex, and various other drugs for prostate cancer and so on. So now I come to my conclusion. The similarities between the two projects can teach us a few things about the process of molecularization, about the importance of feedback loops between theory, R&D. Both projects were very much blue sky projects. They were not seen by ICI as being the pillars of their research, not just within ICI as a whole, but the pharmaceutical section. They were sort of, almost like pet projects of individual scientists, however brilliant they were, or however long they'd been with the company, as in the case of Walpole. They represented a mix of speculative and empirical research, using both existing theories and existing drugs as starting points. They depended on chemical backup and even prowess. I mentioned Dora Richardson's prowess in relation to tamoxifen. But drugs also represented a kind of back and forth between the bench and the bedside, and between theory and its application in the laboratory and in the clinic. So the story I've told you, I hope, presents a much more complex and interesting trajectory than a simple linear process of molecularization. By acting at the level of receptors, ICI's drugs made it possible to better treat, but also better understand diseases such as angina pectoris, hypertension, and breast cancer. Drugs were therefore tools for understanding disease and for a learning process that would lead to subsequent drug design. However, the similarities between the two projects also suggest that big organizations like ICI can become locked into particular modes of therapeutic innovation, which may help to explain why when Black and Jordan left, they missed potentially fruitful avenues for research. And one might reflect on the departure that making vaccines represents for AstraZeneca, and whether that hasn't partly contributed to the difficulties that their vaccine has suffered in the current situation. So thank you very much.
Thank you very much, Viviane, for that wonderful talk, wonderfully insightful talk about how the innovation based on receptor theory and on the use of chemistry was so powerfully taken up by ICI in the 1950s, 1960s, and indeed even the 1970s and later. We've only got one question so far. We may get more later, but it's a question by me, actually. Why did firms like ICI focus on chemical synthesis rather than, say, trying to find natural products or using biological approaches such as vaccines or antibodies? Why did they go for chemistry?
That's a good question. Some of them did obviously go for natural products. And there was a part of ICI that was dedicated to natural product research. It wasn't at Alderley Park. It wasn't at Alderley Park. It was in a separate laboratory. I can't remember exactly which laboratory it was. And there was an issue with ICI research being separated into these different laboratories, which could be scattered all over the country, and a relative lack of integration between them. At one point, the natural products chemistry, if you like, gets brought into the fold of Alderley Park, but in many ways that pattern is already set of thinking. So thinking chemically in terms of chemical structure had advantages, but it could also potentially have disadvantages. The advantages were that it did give a sort of way into receptive theory through this structure-activity relationship, especially once you had the right tools for studying the receptors. But you know, if you want to talk about companies that really focused on natural products, you'd have to look at a different company to ICI. So I'm thinking of, you know, companies that made their name in antibiotics, for example, like Glaxo or Burroughs Wellcome. Mm-hmm. So there's a sort of specialization. Mm-hmm.
I suppose the fact that it was a chemical company partly a clue to the answer in a sense. But, obviously, at a chemical company, it will be inclined to do chemistry. Now, I've got some more questions now. One is, what were the other side effects of pronethanol?
Pronethanol? I think the problem it wasn't very strong. It wasn't very potent, and it was, I think some of the side effects were similar to those of Inderal. It had these effects that were unwanted on other tissues. So in any case, it was going to be replaced by other better drugs, especially once competitors were hot on the tail of ICI and could demonstrate slightly better therapeutic ratio, or so a lower incidence of side effects, or even, you know, more powerful, therefore need for smaller doses. And sometimes there are incremental improvements as well, where you only need to take the pill once instead of several times a day. They were the sorts of improvements that could be introduced, and meant that companies like ICI were forever looking for new drugs to keep abreast of what was going on in other companies.
I had a comment about the lack of biology at Alderley Park from Pete Quirke. I mean, it's in chat if people want to read it, but I'll read it out now. There was an actual product section at Alderley Park in the 1970s, led by Joe Stacey. As far as I'm aware, nothing of any consequence was discovered.
Yes, I think that I remember, obviously I didn't revisit that for my talk today, but I remember the natural products section being brought in to Alderley Park. Yes, the 1970s sounds about right, but it came from outside, and how well integrated within our Alderley Park it was, I don't know. I think part of the reason was that they, you know, was also pooling together big expensive machines like MMR machinery, and to make good use of them, you would start bringing in, you know, bits of research done elsewhere, or bring their instruments and bring them into Alderley Park, just so that the machines could be shared. That was one rationale that I came across for moving the natural products section.
I've had one other comment about Alderley Park, which I'll also read out, which is that from John Waterton, there was significant cancer research at Alderley Park in 1980, leading, for example, to Dolodex or Gosherin.
Yeah. Yes, so that's something that's puzzled me. Thanks for that comment, because obviously the tamoxifen, that ICI chose to continue building on tamoxifen, but I haven't seen any archives or any documents relating to a project like the one I've described. And I've been told that there wasn't really a sort of cancer research program at Alderley Park, but I may be wrong, until, you know, a formal one, until the whole center was redesigned as a cancer research center in 2006, I think. Alderley Park was renamed sort of Cancer Research Center for the company. So who exactly was involved with the research? I think, was it Barry Furr who worked on the drugs that followed tamoxifen? Yes, Barry Furr, yeah.
I've had two comments on contraceptive. One is a question and the other is a comment. One is from our speaker next month, Christine Hodgson. She said, I worked for a wife on a contraceptive pill, but never got to the market. Did ICI ever produce a marketable product? A marketable contraceptive?
As a contraceptive pill, not that I know of, no.
Well, that's interesting you say that, because we've had a comment about that from Phil Gamelin, who said, there was a story at the time, which may be apocryphal, but the chairman was a Catholic and stopped the work as soon as he heard about it. There was work in progress on CPDs that later proved to be of value.
Ah,
and
well, thank you for that.
Who the chairman of ICI would have been in the early 60s. I mean, it used to be people like McGowan and what have you, but in the 1960s, I'm beginning to forget who.
You see, I mean, what's interesting is I've really done this research from the bottom up rather than the top down, which leads to some interesting observations like the ones I've been able to do today. But what's missing is really, for me, the central archive that used to be at Millbank, and goodness knows where that is now, which would give me a top down, you know, sort of, you know, sense of direction from the top, which is something that I don't have as much.
It used to be in a swimming pool, didn't it? It was in the swimming pool of Thames Thames House at one point.
Was it?
Yeah.
I hope it was empty. Yeah.
Anyway, this is a rather technical question from Diana Leach, who of course is connected with ICI herself. Does anyone associated with Alderley Park in the early days have contact with Dr. Fishburne or his descendant, who was one of the chemists who was painted in World War II for an ICI book of employees? I seem to remember some of those pictures appeared in Reader's History, didn't they?
Yes, I don't know.
Can I just say, so, if anybody else has the answer to that, could you put the answer in chat? I'm sure Diana will be grateful. But carry on, Viviane.
Yes, no, I don't know. I mean, it's possible. With the research I've done on the research reports, I came across so many different names. That name doesn't stick in my mind. So I'm afraid I can't help there. Okay.
Somebody from Waterton again says that the follow-up to tamoxifen was the more pronounceable name, fulvestrant. Fulvestrant ring the bell with you? And Barry,
yes, well, my research stops in 1980, partly because of the 20-year rule. I was doing this research in 2000, and this is where the story becomes very sad, that so because of the 25-year rule of accessing archives, and I suppose, you know, the secret nature of some of the research done, or, you know, I wasn't able to go beyond 1980, and the archives that I was able to access as a sort of one-off were eventually closed to anybody from outside. And when Alderley Park moved, when the research centre was closed, they stopped employing an archivist. In fact, I dealt with an archivist who remembered all these people I've talked about, because she'd been working there as a librarian archivist since she was in her early 20s. And when she retired, they didn't replace her, and they just stopped managing the archives in this way. So it's not really possible to continue the story that I've told you. From research reports, one would have to rely on publications, but thank goodness that ICI were always a company that was quite encouraging. Well, they were a company that encouraged publications, although I suspect that would have changed as well later on in the 19th, you know, towards the end of the 20th century, the later decades. I don't know, maybe some of the people who worked there could confirm that.
On the matter of archive, we've had a comment from Diana Leach again. They say, what archive did you use for this talk and where are they? That by we, I seem to mean Catalyst, have the General Chemical Division archive and the Brunamont one as well in Woodford.
So I used the research reports of the Pharmaceutical division, which were kept in the library as a resource for the researchers there, which I thought was really interesting. They would use these old reports to revisit old research, sometimes research that was 40 years old, and they kept it alongside with books and other papers in the library itself. Well, in the library, not on the shelves, obviously they were in storerooms, and they had a card catalogue which I used to begin with, and eventually I used, I think they were starting to computerize it, digitize it, and eventually all the lab reports were digitized as well. And I think the habit of writing these reports may have ended too. So I'm not even sure, even if they opened their archive, whether you'd still have the same type of research reports, which read like, you know, almost like publications or stepping stones towards publications with references to articles and so on, that people like Walpole, heads of sections, would write for meetings with the managers. And it was in fact Alfred Spinks who I believe instigated this sort of way of reporting the different projects once he rose up through the managerial ranks.
So you're saying basically that people might not, if another historian wanted to come along and write about it, they might not be able to get access. Is that what you're saying?
Well, they might not be able to get access. I think because I've lived through the same thing with the archives of GSK, when Glaxo Wellcome was merging with SmithKline and French to produce GSK. They were getting rid again of their archivists, and the whole corporate memory was going with them. And as I was working on, that's another article I need to publish on their antiviral research program, which led to Zovirax and eventually AZT against AIDS. It felt like I was working on boxes and they were waiting as I was working on them in order to put the papers in the boxes and send them into a storage room somewhere by the Thames. And once companies like that lose the knowledge of what they've got in store, they lose their corporate memory, but they, thank goodness there are people who still remember those times and can give oral testimonies about them, but the written evidence is sort of disappearing, and the fear is that eventually they'll be destroyed or be damaged through neglect. I've worked on archives in France where some papers were kept under a roof and the roof was leaking, so the papers got soaking wet. Or another one, there were wonderful papers by Ernest Fourneau, the French pharmacologist, and I worked on his papers in a room of the multi-story car park of the research center. And when I wanted to go back there to look at them, I found the room, and the man who opened the door for me showed me a room with shelving that was empty, and he confirmed that they'd all been incinerated. They no longer existed.
It's interesting you say that, Vivian, because we've had another comment from Diana Lees that says much of the material from Alderley Park was moved to the John Ryland Library a few years ago. I will inquire to exactly what is there, and we'll let you know. I don't know if you mean you or me, but either way, I'll make sure you get the information.
Yeah, that would be very interesting, because I wonder if it was, somebody like Spinks's baby, and by the 1990s, had he retired? Or I do wonder, sometimes that sort of practice is tied to individuals, and when they're gone, that practice goes as well. There's nobody to defend them anymore.
I'm a bit, yeah. I've also had a comment from Phil Gamlin again, saying that the ICI report system was live up to the point where ICI was sold to AKN, Axco Nobel. It was fully searchable on the ICI Lotus Note system, but the trouble is, I believe it's been very, very difficult to get any kind of access to Axco Nobel.
Yes.
So that's interesting information, but I'm not quite sure.
Yes, again, information. It's a shame. I don't suppose that what's in the chat can be kept, can it? You can't save it, can you? I suppose we're recording the talk, so you reading the questions means that the questions and the answers are answered in that way, or are kept in that way, will be recorded. Because obviously there's a lot of information that helps me too.
You want the chat as well? Do you want me to try and save the chat? I think I might be able to save it.
I don't know. I mean, I'm with this Zoom, I'm learning things every day. So, we can. There's a small 3 dots button.
Okay. Somebody says it's a 3 dot button and says save. Right, okay. Right, okay, I've got that. No, no, not that one. I'm going to just try and save it into a file, and then ask my problem is this is a brand new computer for me.
But going back to the issue of cancer research continuing, I wonder, did it continue as sort of product, as development programs within development programs, or actual research projects? I'd be very interested, because I've talked to Craig Jordan, and of course he's talked a lot to me about his side of things, and I'd be very curious to know how, in what form the research continued. So if anybody, or into cancer, if anybody can enlighten me. I mean, there is a book that was produced by somebody called Hill on Alderley Park, but I find it quite difficult to use. It's got loads of little boxes and little asides and so on, so it's quite difficult to use.
Maybe we should leave it there for now, I think.
Yeah,
Viviane.
yes, certainly. But thank you very much for your questions. Very helpful in what is a never-ending project, as you may have gathered.
Yeah, right. No, I'm just I'm just typing something, so 1 second. Yes, well, thank you very much, Viviane. What do you comment here just at the last moment? Phil Gamelin says, many of the ICI divisions put things into local archives. So the organic division report went into the Manchester John Rylands library, so that's the Teesside businesses went into the Teesside archives. Yes, I knew that. The Millbank went under Salisbury Plain. There were a lot of concerns about ambulance chasing lawyers looking for called actions against chemical companies, and DuPont and Dow, for example, deliberately destroyed archives to prevent this. Yes, I know. I believe there's been a lot of destruction of archives all around, really.
Yeah, especially industrial ones.
Well, on that rather sad note, I think we should leave things. And thank you again very much, Viviane. And thank you, everybody, for coming. We hope to see you again next month in May, when we'll be having Christine Hostock talk about dyes this time rather than pigment, from the ancient Egyptian right up to the present. So I'm sure that'll be a fascinating talk, and the NEA alert and my own personal alert will go out soon. Okay, everybody, thank you very much.
Bye-bye.
Goodbye.
Bye-bye.